Okay. So I will go ahead and introduce our speaker. Hi everyone, my name is Avisha Das and welcome to the 116th Stanford MedAI Group Extend session. This week we have with us Dr. Arko Barman from Rice University to talk about his work on deep symmetry-sensitive networks for detecting brain diseases. Dr. Barman is an assistant teaching professor in the Data to Knowledge or D2K lab at Rice University, and he's affiliated with the departments of EC and statistics, also at Rice University. He received his PhD in computer science from the University of Houston, and he has also previously worked at the Palo Alto Research Center as well as at UT Health and Broadcom before joining Rice. He is also actively involved in data science education research and has published and presented and has been in the organizing committee of different engineering and computer science education conferences like ACM6 and IEEE Frontiers in Engineering. Dr. Barman, thank you very much for joining us today. And before we start, do you have any preferences on how you would like to take questions? Like, is it okay to interrupt, or do you want to keep the questions till the end of the session?
I'm okay to take questions in between.
Okay.
I think that aids in understanding if you're stuck, probably.
Okay. Yeah, I mean, perfect. So let's try to make the session as interactive as possible. And without further ado, let me hand it over to Dr. Barman. Thank you.
All right. Thank you, Abhisha, for the introductions. I don't think I need to add anything to it, except perhaps that this is a project I was involved in, which I'm probably tangentially involved in, but not directly as I was some time ago. All right, so let's start. First, I will have an overview of what I'm going to talk about. So first, why do we need symmetry and how it appears in our organs and how we can leverage that symmetry to detect diseases. And then I'm going to introduce deep symmetry-sensitive networks. It's basically a paradigm, and it's sort of like you can consider it as a family of architectures that you can tweak and design yourself. Then I'm going to talk about the application of DeepSymNet for large vessel occlusion in the brain, and how we can also interpret the decision-making process of DeepSymNet, so it's also explainable. Then I'm going to talk about an expanded version of DeepSymNet, which combines both symmetry-sensitive features as well as symmetry-naive features, which is basically what we are used to, just like a network for detecting brain hemorrhage. And finally, another application of DeepSymNet for quantifying neurodegeneration in Alzheimer's. It doesn't technically use symmetry, but it's a similar kind of network, and we're going to talk about that as well. So first, let's talk about how we can leverage symmetry for detection of diseases. In a lot of diseases, there are organs, and these organs have some form of symmetry. For example, you can think about prostate. Prostate cancer is usually detected or diagnosed using MRIs, and there's an element of symmetry in the prostate. You can think about detection of breast cancer using mammogram. There's an element of symmetry over there as well. And finally, brain diseases, they definitely show some patterns of symmetry and how deviations from symmetry can be indicative of diseases. However, it's necessary to emphasize that in our organs, in our body, symmetry does not mean exact mirrored copies. It's sort of like a quasi-symmetry or like an approximate symmetry, but not an exact symmetry. And while building models, we do not stress on an exact symmetry, but like a quasi-symmetry or an approximate symmetry. So one of the popular paradigms in designing neural networks is inspired from Siamese twins, and it's called Siamese networks. So if you're not familiar with Siamese networks, it's basically a model, a deep model with the same structure, and you might have two different inputs going into the same structure, and what comes out of these two models is sort of a representation, feature representation of what goes in. And then you can calculate the difference between the feature representations to get an output. Now, this output is indicative of whether these two inputs are similar or not. So in the traditional Siamese architecture, there is a loss function which is called contrastive loss. And it measures the distance between what I said was like the feature representation of the two inputs. Now, if the distance is less than a certain threshold, then the inputs are considered similar. So which gives us an element of approximate symmetry, but not exact symmetry like I mentioned before. So you can have a threshold or like a sphere, and any distance lying within that small sphere is going to be considered similar. Otherwise, they are dissimilar. Of course, this kind of a design has its own limitations. For example, it might not be suitable for all types of data. And definitely there is some limitation with the use of a loss function. Loss function is something that you and I define, but the data might not suit the loss function. So instead of having a loss function, can we replace it with something else, which can also be learned? So there is a learning process when we compare the two feature representations. So in the case of DeepSymNet, what we did was say you have a radiological image, and this image is, of course, from an organ which has some sort of a approximate symmetry. And you can split it into a left half and a right half, for example, or in any other way. And you can flip one of them so that they kind of look similar. And then you pass them through a network. This could be any kind of a network, and we're not talking about its design at the moment. And then what we do is we take the absolute difference between these two features. And finally, we have another CNN, or it could be, once again, any kind of a network in reality. And that is going to use this absolute difference between these two and calculate a symmetry-sensitive output, which basically learns from the symmetry. And if they are very similar, then this L1 difference is going to represent that. But notice that this is not a number, but what comes out of this is actually a feature map. So you could use those feature maps to understand the differences in symmetry between the two inputs. So now, how do we design these two kinds of CNNs? One is a representation learning CNN, which learns representations of the two inputs, and there's also the symmetry learning CNN, which learns features related to symmetry. So we
Sorry,
don't.
Arko, can I ask one quick question?
Yes, please.
So can you go back to your diagram once?
Sure.
So in the symmetric learning CNN, you are feeding the absolute difference only, or you are also feeding the learned representation?
So in this network, in this paradigm, we used an absolute difference between the feature maps.
Okay. Okay. So you are just taking the absolute difference in the feature map, which is not like a one single value. It's again a metric.
It is potentially a tensor because it's a
Okay,
feature map,
perfect.
right?
Okay. Okay.
You could also actually concatenate them if you wish.
Okay.
And in that way, it's not strictly a symmetry learning procedure because you're not looking at differences, but it's sort of learning the differences in some other way.
So you actually flatten them, right? And then calculate flattened or the
No,
absolute difference, or it's originally the actual dimension metric.
it's the feature map. So it's like a
Three
tensor.
dimension.
Yes.
Is it three dimension, two dimension, one dimension?
It's technically of 4 dimensions
Okay.
because the feature maps are 3-dimensional because
Okay.
this is a 3-dimensional radiological
Okay.
image.
So you're not even taking like 2D images. You are actually directly feeding the 3D volumes.
Exactly. So
Okay.
these are 3D volumes, and then you have, of course, filters. So you have 4D
Okay.
maps at this stage.
Okay. So your symmetric learning CNN structure is different because that is like 1 dimension more compared to your representation learning CNN. 1 dimension more input, right? So your representation learning CNNs are 3-dimensional CNN, while your symmetric learning CNN are 4-dimensional then.
Yes.
Okay. Okay.
All right. So if you want to design this, there are no restrictions in designing. And while I was designing it, I was sort of, this seemed very funny to me. And if you're from those ages, like early days of deep learning, there was this popular meme where, it's not enough, you have to go deeper, from this movie Inception, which actually inspired the name InceptionNet, if you're familiar. So the InceptionNet was actually inspired by this meme. And it actually looks like this. So it basically has blocks or modules. And these modules were actually 2-dimensional. We made it 3-dimensional. And it captures different scales and different sizes of convolutions in different ways. So there are 3 times 3 convolutions, 5 times 5 convolutions, and also like 1 times 1 convolution across filters, and so on. So it basically captures different kinds of features in a single block or a single module. So based on the complexity of the problem, you can keep stacking them if you want. So if you have a really complex problem or an organ or a kind of imaging process which gives you really complex images, you can keep stacking them. For mammograms, we saw it needed a very large stack, but for brain imaging, it needs a smaller stack. But that's sort of empirical. And then, of course, there's these differences, and then you can again keep stacking them and learn symmetry. And finally, of course, you're going to have a max pooling and a fully connected layer, and that gives you the result of the network.
I have a question. Oh, sorry.
Yes,
If you
good.
So the number of stacks that were needed in the, like, are the number of layers in your representation learning CNN the same as the number of layers of IM or inception modules required in your symmetry learning CNN? I can understand the 2 CNNs in your representation learning, like the two modules on the top and the bottom, they are the same, but is it also consistent in your symmetry learning CNN, or you can play around with the number of
Yeah.
layers?
So like I said, it's empirical,
Mhm.
and you actually need to run experiments and see how deep it needs to be.
And is there like a trade-off between the computational complexity and the number of layers that you're using, or is there something that you have seen that with the using like larger, say for example, like you mentioned for mammogram you need like larger number of modules, and for brain you need like a smaller number of modules. Like, is there a set number that varies, or is there a trade-off somewhere, like where you just kind of fix on a particular number because you're not seeing any improvement beyond that? Like, how do you, like, is
Well,
it all empirical? Like, how many experiments do you have to do, or
yeah, it's sort of empirical, but I can give you some perspective. Um, for brain, it's a 3D volume, and it's a very dense information source, whereas mammograms are 2D. And personally, I don't think that's like a hugely dense information source as much as brain imaging. That's perhaps one of the reasons we need to have deeper networks.
So Orca, for mammogram, did you resize the image then? Because mammogram images are huge, right?
Yeah, all images were resized
So
resized
don't you
into
think
a
that you would lose some information if you resize, particularly in this case where you are comparing right versus left?
So in
Because
case of these brain images, they were resized to a really, really small size compared to what a CT gives us.
so CT gives us 512 by 512. So how did you resize it?
Um, our CTs were actually far larger. It was about 3,000 times 3,000 times, number of layers, which were like eight slices of the brain. But that was
Only 8?
yeah,
So what was the protocol? Do you remember? Like it was actual, sagittal, it was with contrast.
it was CT angiogram, so of course it was with contrast.
All right. So ideally CT angiogram slice thickness are like very small, like 1 millimeter, 1.5 millimeter. So usually you should have more slices. Not sure why
Yes,
you have only
I
8.
yes. So I'll take that back. So 8 was what we reduced it to.
Ah, okay. Okay.
The
Okay.
actual was in the hundreds, I think.
Yeah, it should be like more than
Yeah.
150.
Yes, I'll take that back. So in our scaling down everything, we scaled the slices down to 20% of the size of the actual image, and we reduced the number of slices to 8.
So do you know which device you are using? Because I have never in my life of like 15 years, I have never seen any city that dimension is 1,000 by 1,000.
That is something I do not know, but it
Ah,
was in
okay.
the thousands. Yes.
Okay. Okay.
But we also actually did registration and stuff, so
Mhm.
maybe that changed the sizes a little bit
Okay. Okay. Because
because,
mammograms are usually more than 1,000 by 1,000. Like
oh yeah,
can even
those
go
were,
to like three,
yeah,
2,000 by 2,000,
yeah, those were really huge. But what I would like to stress on is that even if you reduce the size very aggressively, even then the model performs really, really well.
I didn't understand that. What was your resizing like size after you resize it? What was your resolution?
So off the top of my head, I do not have those numbers. It's there in our
Okay.
paper, of course. Um, but what I do
I
remember
know it.
is we resized every slice down to 20%, and we had only 8 slices.
Okay.
Yeah, the details are in the paper, of course. Feel free to send me an email. I can send you the
Sure.
papers.
That would be great. Yeah. I
Yeah.
don't know, Vishal, you already have the paper or not.
I don't. Yeah, I think I might have it. Yeah, I'll
Okay.
share.
All right, I think that was a good question. The bottom line is, even if you aggressively um, downsample or interpolate and make it smaller and smaller, even then the model performs really well. So since there are no networks of this kind, we had to actually come up with benchmarks because we didn't know what to compare this to because this was such a novel idea. Um, so we had a symmetry-naive CNN, which is basically like a CNN where you keep stacking the layers. We had a traditional Siamese network, which used contrastive loss to learn differences. And another thing that we also did was we used the pixel-wise absolute difference between the two hemispheres of the brain or the two halves of the image and just put it into a CNN and see how it does. So now I'm going to talk about the diseases a little bit. First, um, we use this for detecting large vessel occlusion. Large vessel occlusion is actually a subcategory of ischemic stroke, and ischemic stroke is actually the most common of all kinds of stroke. For everyone who's not aware, it's basically um, a blockage of a blood vessel, and it consists of around 87% of all strokes. It's really deadly, and the faster it gets treated, the more chances you have of surviving or avoiding a debilitating sort of condition where you cannot move around or have like physical disabilities because of the stroke. The other big thing is there is a very high mortality within 3 to 6 months, even if mortality doesn't occur really quickly, it might occur over time. And it's actually about 20 times higher than people who do not have a large vessel occlusion. So basically, a large vessel is occluded in the brain, and that leads to cascading problems. So what the imaging technique we used was CT angiograms. CT angiograms are widely used, as far as I'm aware, for detection of large vessel occlusion and ischemic strokes in general. Here are two sample CTA images of a control patient and a patient with large vessel occlusion. If you look at it closely, you'll see for patients with large vessel occlusion, you can see these blood vessels. These blood vessels are bright over here because you have a contrast, and the imaging sort of, you know, makes this contrast looks brighter. So the other half of the brain looks relatively dark, which means there is a lack of, blood flow in the opposite hemisphere, which actually shows you that there is an element of asymmetry caused because of the disease. So one half of the brain is receiving blood, whereas the other half is not in certain regions. So I just blowed it up a little bit, and if you look at one of these images, you can see these are blood vessels on this side, and on the opposite side, there are dark regions, and these are the regions where blood vessels were supposed to be there, but they are occluded. So like I said, it just shows you that the two hemispheres look different because of this disease. So the disease can be detected using some sort of a network which catches differences in symmetry or like deviations from symmetry, basically. So our data set had 195 control patients and 250 with LVO. And these were our results. These are ROC curves, and the area under the curve for DeepSymNet was about 0.88. And we also did some permutation testing and got some confidence intervals, and it went as far high up as 0.92. Okay, there are some important and interesting aspects. One is learning from smaller data sets. As we're all aware here, training samples are really scarce in the medical research community. Data sets are getting larger, but still not like really large. And you can also say like our data set was not really large. It was just about maybe 450-ish. So the question is, can this model or this paradigm actually handle smaller data sets? So what we did was we used progressively smaller data sets and looked at whether this model can perform well, even if we have smaller data sets to train it on. It seemed that it was we could reach about 0.8 area under the ROC curve with as few as 50 training samples. So which clearly shows that there is a potential for using this model for data sets that are actually small in size. We also looked at an interpretation. We do not trust black boxes to make decisions about health and treatment. Of course, I wouldn't trust a black box, neither would you. Of course, clinicians and caregivers also do not trust decision-making processes that do not have an interpretation. So I strongly feel that interpretation is an important component of any computer-aided diagnosis or computer-aided treatment process. So what we did was we used an explanation technique called epsilon LRP to look at which pixels or which voxels, technically, which voxels in the images were most significant in the resulting decision. And we noticed that if you look closely at these activation maps, these regions of the brain seem to have a really high activation, whereas the corresponding regions of the brain in the other hemisphere have really low activation, which points out the differences in vasculature. And we actually showed a lot of these activation maps to clinicians, and they sort of agreed at a qualitative level that yes, these are actually the regions that they would look at in the image to actually detect whether a person has LVO or not.
Arko, I have a question here.
Yes.
You're back propagating to your model has like 3 different modules, 2 representation learning, and then 1 module that learns from the difference between the presentation. So how far back are you back propagating for this explanation? Up until the input or up until the module where the difference is computed between the left and the right hemisphere?
Yeah, the interesting thing is we're back propagating right from the last layer back to the first layer, back to the inputs. So if you remember, we did a absolute difference of feature maps at one point, but that didn't stop this method from actually going back to the inputs and telling us which voxels in the inputs were contributing. And the nice thing is we were surprised it worked, because we're losing information about which hemisphere has a stroke and which hemisphere doesn't when we're taking the absolute difference. But when we look back at these activation maps, we can actually see the correct hemispheres having the vasculature, which means blood vessels, and the correct hemisphere that doesn't have blood vessels showing up.
That's interesting. So because you're feeding left hemisphere as such and then flipped right hemisphere, so I'm assuming that just for the representation purposes, you flipped it back to the original position and then you're showing these images.
Oh yeah, that's
Okay.
like a post-processing, like
Post-processing attempt, okay.
Yes.
Okay, makes sense. Thank you.
All right, so some observations that we had, which we felt was interesting. DeepSymNet actually learned symmetry in vasculature in this problem, which is exactly what we were targeting. And the anomalies in vascular symmetry that it learned was similar to the diagnosis by clinicians, and the interpretations were also similar to what clinicians actually look at. We also looked at statistically significant activation in these activation maps and found these red regions where we believed there were a lot of vasculature, and the corresponding missing regions in the other hemisphere where we noted a lack of vasculature. Okay, quickly moving on. I don't know if we are keeping to time, but I would like to discuss a couple of more applications of this kind of a network. So this was the network that we started with. So we had 2 hemispheres of the brain. We extract representations, then we get an absolute difference, and then we had a symmetry learning network. So this absolute difference actually forces the model to learn from symmetry only, but symmetry is not the only mechanism for detecting diseases. We also look at the entire organ as a whole, right, to understand if a disease is there or not. So we thought that, okay, how about we tweak the network a little bit and see if we can combine both symmetry as well as an overall picture of the organ for detecting diseases. So the next idea is where, excuse me, we have representation learning CNNs with the exact architecture and the exact same weights. And we do calculate symmetry features over here using absolute differences, but then we decide, okay, let's concatenate them and we can look at an overall image as a whole instead of just symmetry. And we concatenate them and we use a CNN to extract features or learn features, and we call this a feature fusion CNN. So this feature fusion CNN actually sort of gets a fusion information from both the symmetry features as well as the symmetry-naive features in a way.
So in this case,
Yes.
you are both feeding in the absolute difference as well as the representation feature vectors, like the feature
Yes.
maps, right?
Yes. Yes. So the raw feature maps as well as the absolute differences. So we chose a disease where, of course, we had the data, and also we thought that the symmetry might play a role, but overall, in the detection of this disease, clinicians actually look at the entire organ instead of simply the symmetry. So brain hemorrhage is possibly the second largest category of stroke, and it affects millions of people worldwide annually, and it has higher mortality and morbidity compared to any other subtype of stroke. So in any automated stroke detection method, we actually need to detect whether brain hemorrhage has occurred, because we want to administer the treatment very quickly. And so it also helps us distinguish whether the patient has ischemic stroke or not, or like a brain hemorrhage. And actually, in the first stage of diagnosis, it's more important to detect hemorrhage rather than the segmentation of the lesion, because we want treatment at the acute stage. Later on, perhaps we want segmentation of the lesion, but initially we actually want to detect whether it has occurred or not. Now, as you can see from this image, it's a non-contrast image, and symmetry might or might not play a role. I mean, in this case, it perhaps does, but the lesion can be in any part of the brain and it might have nothing to do with symmetry at all. So our data set was a bit complicated. We had about 568 patients. Some of them had stroke, some of them did not have a stroke. And out of the patients who had stroke, some of them did not have brain hemorrhage and some had brain hemorrhage. So what we did was our controls actually had patients who had no stroke, plus who had stroke but not brain hemorrhage. So there were like confounding factors in a data set which are likely to make the model more confused, basically. So it's not just healthy people in controls, it has people with stroke, which is not brain hemorrhage. Without looking at the legend, I mean, it compares a lot of different combinations of DeepSymNet with other stuff. The purple curve actually shows the ROC for this expanded DeepSymNet model. And it was almost near perfect. And it actually outperformed DeepSymNet by itself. DeepSymNet also actually performed pretty well, I think. So which actually shows that it's not just symmetry, but overall, the anatomy of the organ itself overall is also needed for understanding the disease. So this made us think what happens. Okay, before that, once again, we looked at the activation maps and we saw these yellow spots which lie inside the lesion actually, which tells us that yes, the model is actually looking at the correct regions in the images. So it actually, this whole thing made us think, okay, so what is the contribution of symmetry versus symmetry-naive, meaning like the entire organ features? And in this disease, I mean, it will, of course, be disease dependent. We noticed that the highest activations were because of these features which were sort of looking at the entire organ, as opposed to symmetry features which were in general less relevant towards the prediction of the disease in this case. But of course, it's going to be disease dependent. All right, finally, I hope I'm sticking to time. I'm going to talk about neurodegenerative progression. So for diseases like Alzheimer's and Parkinson's, which are neurodegenerative diseases, there is a sort of a steady decline in how a patient does, right, over time. And if we do a longitudinal analysis of radiological scans, we can somehow analyze, understand, and quantify how worse the patient is getting or how fast the disease is progressing, right? So in this case, what we can do is we can take two brain images at two different points in time and use those two as the inputs. So in this case, we're not looking at symmetry. We're looking at the entire brain volume at two different points in time and seeing how they have changed, right? So we can look at brain volumes of the same person who's got Alzheimer's in two different points in time, or there is a case where people slowly progress. So from control, you move to an intermediate stage called MCI, which is mild cognitive impairment. So you have some cognitive issues, but you haven't reached Alzheimer's diagnosis. So you could have like one brain volume when a person was MCI versus when that person progressed onto Alzheimer's. And you can train your model. So in this case, the goal of the model is not to, I mean, of course, it's trained on whether it can classify progression. But the goal is to understand how progression happens and quantify it rather than on the actual prediction or the classification output. So in this case, as you see, we have a brain volume and we can look at the activation map and see which regions of the brain have progressed more in the disease and which have not. So we can understand which parts of the brain have like more degeneration, like tissues in which part of the brain have degenerated more and which regions have not so much. So this is an image of the whole brain, and the brighter the region, the more it has progressed degeneration for the disease. And you can also split it up using cortical and subcortical regions of the brain using an atlas. And what you're going to see is what we saw is the subcortical areas have like more progression over time, like faster or like basically more progression over time. And the other interesting analysis is we can split up the entire brain volume into smaller parts and look at which parts, not just at the cortical or subcortical level, but which smaller parts of the brain have progressed more because of the disease. So for subcortical, we see like some of these regions have progressed more, others a little less, and same for the cortical region. All right. In conclusion, we can see that leveraging symmetry in model design, we can often get better performance compared to a simpler network. It is also capable of learning from small data sets, and interpretability actually shows us that what the model looks at is actually the correct regions that we wanted to look at based on how clinicians make their diagnosis. And finally, we can also use this network or this kind of a paradigm to compare not just two halves of an organ, but like the same organ over time and look at diseases like Alzheimer's, where you can see progression of a disease over time. These are the papers in case you're interested. You can also sort of send me an email and I can send you the PDF versions if you cannot access them. That's it.
I just would like to thank you so much, Dr. Barman, for your talk. And please, if anybody has any questions.
Yeah, Dr. Barman, thanks for the great talk. One quick question: the CT brain hemorrhage task.
Yes.
There was a like I think RSNA had a competition maybe 2-3 years, maybe 3-4 years ago, but there they were trying to, because the brain hemorrhages have different subtypes. So I think
Yes.
there are like 5 or 6 subtypes. So the challenge was to classify brain hemorrhage on those CTs and like into those 6 classes. I remember a lot of us were like trying like a 3D approach, but what really surprised me back then was like almost all the people who got the best results. The idea was that they ran 2D CNNs on each slice. So
Yeah.
then you run 2D CNNs, get a feature vector, and then once you have a set of feature vectors, you feed it to some RNN or at that time even a LSTM. And that combining 2D CNN and LSTM was like so much better than the 3D CNNs. And just curious, like I can send you the links. I can put them in the
Yeah,
chat. It'll
sure.
be interesting to see how this 3D approach and the data set is well defined. It comes from RSNA, so it's a pretty big data set.
Yeah, I mean, a lot of times, just looking at the slices are enough to tell you how the disease looks like. Um, um, for brain hemorrhage, I think that's a really feasible or practical thing to do. But I'm not a clinician, but perhaps that's sort of not the best for
Yeah.
large vessel occlusion, because you want to see how the vessels sort of take shape in a 3D volume.
In 3D, yeah. Okay. Yeah, makes sense. Yeah. Thanks.
I had a question. So
Yes.
for the neurodegenerative or progression, so did you also measure any rate of progression, like how fast the different parts of the brain were like degenerating, or like was there any other component where you kind of quantified the rate of progression as well?
So what we did was quantify the progression of the different parts of the brain,
Mhm.
but not really at a patient level, right?
Okay.
And that's actually also not very well defined in the clinician community, right?
Yes.
Because there are stages, and those stages are sometimes pretty fuzzy or overlapping. So like if there's a stage 1 and stage 2, some clinicians might tell you you are in stage 1, some clinicians might say you're in stage 2. And there's, as far as I understand, there is not a very clear demarcation of how these stages happen.
Yeah.
Mostly these stages are also defined by some cognitive tests rather than imaging.
Okay. So there's no clear way of kind of putting like labels as to, okay, at this stage, this is going to be the, like, this is the image and this, so that you can, and it can change, the rate of progression can change. So
Yeah, for
it's
example,
not
a
from
person
patient to patient.
can be an MCI, like mild cognitive impairment, throughout their life without
Mhm.
ever progressing to Alzheimer's. Or
Mhm.
people might go to Alzheimer's and then stay at the early stage for the entire life, for example. Or they
Okay.
might progress slowly, they might progress quickly. And it's very difficult to often sort of understand. Also, one thing is these MRIs are expensive
Mhm.
and they're not done for every patient visit. So we do not have like a time scale or like, okay, these MRIs are 6 months apart for every patient or something, which makes it more difficult to sort of try and quantify at a patient level how they've progressed. So instead, you can look at how different regions of the brain has progressed through the neurodegenerative disease.
Okay. Okay. Makes sense. Thank you so much. Yeah. So are there any more questions? Let me look at the chat. Yeah. So yeah, I mean, so I guess there are no more questions. So please, I would like you all to have a virtual round of applause to thank our speaker. And with that, yeah. So thank you so much, Dr. Bowman, for your talk today. And
Thank
if
you for
anybody
having
has
me.
any, yeah,
Oh,
and if anybody has any more questions, they can always reach out to him and contact the speaker directly. Thank you so much.
all right. Thank
Yeah.
you.
Bye.
All right. Thank you.
Thank you so much, Arko.
See you. Bye-bye.
Yeah, see you. Bye.